So it's my distinct pleasure to introduce our keynote speaker. Prather is a distinguished professor and chair in atmospheric chemistry at the University of California, San Diego. And she's been kind of a voice of reason throughout much of the last couple of years, thinking about the pandemic and response. And so please welcome Kim to the podium.
Okay, well, it's my honor to be here, and thank you for the invitation to come and speak. I'm looking forward to the next couple of days, and it's wonderful to finally see many faces that I've only seen in little screens, and now there's whole bodies. It's awesome. But today I'm going to talk about sort of it is cross-cutting, I think. It sort of touches on all the different aspects of what Peter was talking about. I basically just sort of to emphasize, I have been actually for NSF. NSF has been very good to me for 32 years. I have directed another center called their Centers for Chemical Innovation. That's another one. And I've directed that for 13 years. So that's CASE, and that's sort of winding down. And that was really focused, as you'll see, I'll talk a little bit about it to give you the context of my background and how the heck I went from being an atmospheric chemist to a person who tracks infectious disease in the environment. They are connected in the end, and I'll come to that at the very end. We've actually spun off another center as CASE is sort of the ocean climate part is sort of the CCI part of the funding is winding down, and we've received a major gift, Romeo Morrow and I, for looking at tracking infectious disease, airborne infectious disease in a changing climate. And so I will talk about sort of all of this in little tidbits, and you can see how they're all connected. Okay, let me see. It's gonna go. Oh, pointing at you. Okay. Yeah. Oh. Was it just slow? It might have been. I might have been impatient. Okay. All right, so this is the center, is to say that I've been directing. It's a large center, and I wanted to put this up here to say that, you know, this center has really, honestly, it changed my whole career. As professors, we're not really educated in how to lead something so grand. It was $40 million, and you know, but I think it helped me learn to become more of a leader. And there's 12 institutions that are involved. It was a huge learning experience, as I say, just from that standpoint. But one of the other things it taught me was the importance of interdisciplinary research. That's the beauty of a center, is that there are biochemists involved, microbiologists, oceanographers, infectious disease people. You know, like it was this incredible mixture of people. With the center on chemistry, but because it comes from the chemistry division, but it really spanned across many, many fields. And we also, the really cool part is being able to train students in all those areas. And so it's been a wonderful part of my career. And I just, it wouldn't be standing here if it wasn't for CASE. So this is sort of the figure that I like to show. It's sort of what got me sucked into being a voice during the pandemic. It's what I was doing when the pandemic started, which was looking at how aerosols influence clouds and climate, in particular, how the ocean aerosols influence clouds and climate. And so as you can see, aerosols travel all the way around the globe. They can be released on one side of the earth, and it takes about 2 weeks to go all the way around. They get incorporated into storm systems and clouds. If there were no aerosols, there would be no clouds. So at the center of every cloud drop, at the center of every ice crystal is an aerosol particle. And I developed an instrument early in my career that lets you in real time look at the chemistry and size of individual aerosol particles in real time. And we've flown that through clouds and looked at how and why clouds form, how and why clouds rain or snow. And so it's been, that's where my mind really was when the pandemic started. But one of the things we had proposed to do to NSF, I'd done, I guess, about 20 years of field studies with these instruments, different instruments. We go out in the field for a couple of weeks. And the thing about field studies is you kind of get to play the cards that are dealt to you. And you have no control. And I like control. And so we proposed to NSF that we were going to move the entire ocean, atmosphere, biology, waves, everything into the lab, where we could actually study the ocean, because the ocean, we know a lot more about terrestrial processes than we do about the ocean, yet the ocean covers 3/4 of our planet. And so what we, so I said to NSF, let's just move the whole thing in the lab where we can control everything. We can control the water, the biology, the temperature, the waves, the bubbles. We can control the whole system. And we can perturb that system and then measure the response. And so again, being chemists, what we really wanted to do is, I'd been studying heterogeneous chemistry my whole career, like how reactions happen on the surfaces of particles. So what we really wanted to do was we wanted to generate sea spray that looks just like the real world, because what people have been using as proxies for decades was salts. But in fact, the ocean is full of life. It gives us over half of the oxygen we breathe. It's got everything, present for life. And so how do you get that in the lab and replicate the transfer processes to get things out? Because I didn't want to just make something. I wanted to make what really happens in the real world, which actually took us 3 years to do. And so, push the wrong one. All right, so I love bubbles. I've fallen in love with bubbles. Oceanographers taught us that we were doing it all wrong as atmospheric chemists. We would take the little frits you put in aquariums, and they make lots of little tiny bubbles, and that gives you lots of spray, but it doesn't look anything like real sea spray. You have to have the actual full bubble size distribution. And by that, I mean, it has to match what a breaking wave looks like in the open ocean. How do you do that? Acoustics and imaging. We actually count the bubbles and size the bubbles. And when you get the right bubbles that look just like the ocean, you start to see really interesting things. And so this is a crashing wave in our newest system, which was also funded by NSF through the MRI program, which is a wonderful program for instrumentation. And this instrument is an instrument that produces sea spray. And if I can get this to go, the mechanisms that are key are this film rupture up at the top, and basically you form a cap, a stable cap. When it ruptures, you see the sea spray break off, and then you see the jets shoot up out of the middle, which sample a different part of the water. And when you do it correctly, you start to see amazing structures come out. So this just shows, bacteria being invaded by a phage. It shows a droplet with a bacterium inside, and these little vesicle type structures that look rather cellular. And so we were really excited when we started to see this. Nobody had ever seen this before, because basically, you have to have the whole transfer process correct. And so we've spent a lot of time understanding why certain microbes get out and certain ones don't. Only about 20% of them get out. And when they do, they get out really well. But as I'll talk about towards the end, things that we've traditionally thought of as waterborne pathogens actually can be airborne. And so that's opened up a whole nother can of worms for us in our research. Speaking of which, one of the, I don't know if I can you click that to get the movie going? I don't know if I have the ability. It's down on the bottom of that. Yeah, if you go down, it should pop up if you just click. Yep, there you go. So this is one of the methods. We developed all these tools. We have wave channels. We have a wind wave simulator. And then we made these little smaller systems for looking at smaller or more fragile samples. So you can see the water spilling. Believe it or not, that gives us the right bubbles as well. And we took a particular, it's basically a mimic for cholera that's in the ocean. It has very similar cell properties. And so by that, I mean, it's got similar hydrophobicity. And we looked at whether it got out or not. And again, basically it got out with nearly 100% efficiency. And this is a mimic for cholera. And so we were actually able to grow it on the plates, which means that it remained infectious even when it was in the air. This is basically something that Rita Caldwell, built her career on. And she's super excited by these results. And we're working with her now to think about, when I first met her, I said, they say cholera is waterborne. How do we know it's not in the air? She said, we've never looked. And so, now we are starting to look, as I'll talk about towards the end. All right, so this ties into the first one of our areas, which is the mechanism of how things move around. And it's a long story. And a number of you have commented. I was very active on social media. As I said, I took a lot of hits for the team. I have been pretty relentless. Tony Fauci calls me a aerosol activist, because we've pushed really hard, a number of us that many of you know. It was a real true team effort, nothing I thought I would ever do, but we put a lot of pressure on public health and many other people, just trying to get people to realize that this virus is in the air. And so I'm just going to talk a little bit about like that bit, just sort of go through the basics of the, sort of how I got to the point where we are now. So the way I got initially sucked in was a person from the news asked me if I thought the aerosols could go further than 6 feet. And so this is that video comes to mind, and I laughed. And as they say, the rest was history. I mean, I think I did over 1,000 interviews in these last 4 years. It was, and many others did as well. It was really crazy. But the other thing I did was I wrote, I don't know why, I guess we were locked down and I had time, and I decided to write this perspective. Never done this before. And it wasn't my research, it was everybody else's research. But to me, it was obvious how this virus was sneaking around the world. And so I decided to write about it. And I contacted Holden Thorpe at Science, and he said, sure, we'll work with you. And so I spent a few months writing this article, which was a perspective, it ended up being a perspective, came out in print in June 2020. I learned a lot writing it. But what the interesting thing is, this basically just said it's being spread by people that don't have symptoms. And so, basically it's just sneaking around, and it has to be airborne. So I gave all the reasons it had to be airborne, and we knew at that time. And so this paper, Holden told me that it's been downloaded in the first 2 months, it was downloaded 1.6 million times. And so that's good news and bad news. Sounds good, but that means people read it, and that means people knew, but they continued to deny that it was airborne, which was just mind-boggling. WHO, this is still a tweet of theirs. It's still up there. It says basically COVID is not airborne. And so I have dealt with WHO more times than I would care to admit. We've had all kinds of conversations. They just really don't want to take down this tweet. And, basically it's led to this misinformation about how you protect yourself. And millions of people have died, and they still won't take down, and they still will not clearly say the virus is airborne. I mean, they have it kind of buried. They snuck it up. December 22nd, 2021 at 11 p.m. at night, they snuck up something about airborne finally, but you had to look for it. You had to know to go look for it. And so, it's just crazy confusing the poor public. COVID, they say COVID isn't airborne. This is an article in Wired. Coronavirus isn't airborne, but it's definitely borne by air, right? And so it's just like, it's been frustrating. So I was involved in a number of ways. I tried every lever I could think of. Okay, this way. Oh. It's not liking me. Like pointing it there, it was working fine. Are you able to? There it is. Okay, this is you can just click if you want. So Science was really supportive of us, and every time I would sort of come up with a different thing I wanted to say, Science would review it. And so the second one was a letter that I wrote. We wrote. It was with an infectious disease doctor. At the time, I knew better than to just go in as an aerosol scientist and talk about medicine. So I got a really good he was the editor-in-chief of clinical infectious disease, helped me write this. And this was based on an actual National Academy workshop where we all agreed that the virus is airborne. And this was August of 2020. Yeah. And so basically the tricky part was that there's this 5-micron thing you hear about size, right? And I even have it on our review that we wrote. We basically said, and this is where Tony Fauci's eyes, I was the one that convinced him that it was airborne. I showed him the physics of how far particles up to 100 microns can go, a lot further than 6 feet, right? And so his eyes popped out of his head, and basically, this paper came out, and I had shared it with Tony, and then I guess there was an embargo on it. It was a short letter just saying 5 microns is wrong, and it was going to come out at 11 a.m. on October, I can't remember what day, but basically, CDC finally said it was airborne at 10:40 that day. So it put the heat. They knew it was going. They wanted to be the first ones to say it. So it served its purpose, at least at that time. And then we wrote this review, which said, it's not just SARS-2 that's airborne. They're all any respiratory. It shouldn't be crazy thought that every respiratory virus has an airborne component, right? And when it's airborne, a lot more people get exposed. I can't think of a way that we would have a pandemic if it was on surfaces, right? It's been ruled out for this one anyway. And that, I mean, I'm not to say I'm not an anti-hand washer, but I do think that that's been overemphasized. And CDC has shown or stated that you have less than a 1 in 10,000 chance of catching it if you touch an infectious surface and go to your mouth, like less than 1 in 10,000 for that virus. Now, other viruses might have more. So I explained it to Tony in a Zoom meeting in August, and then he suddenly became a believer. And so this was just a talk he gave after that, sort of saying, okay, I guess aerosols are really important. And he became, for a while, a pretty big spokesperson on this particular topic, which was great. So getting back to the question of how do I go from studying, and why was it obvious to me that this had to be aerosolized? And, what does the surface of the ocean have to do with the surface of your lungs? And I would argue almost everything. It's very, very similar. So basically, I've shown here breaking waves and the components that are there, and some beautiful modeling work that my co-director, Romeo Morrow, is doing on billion atom simulations of what these aerosols, sea spray aerosols, look like. And they don't look just like salts. As you can see, those are actual lipid islands. And it's just, she's done some really beautiful work. But she's used biology tools, computational tools to do this. And so we've gone back and applied it sort of to the real world. That's the ocean surface. And this is what the work she's done on respiratory aerosols look like. Again, components are almost exactly the same. And guess what? Bubble bursting happens in your lungs too. And so when you speak, you sing, you yell, you breathe, you create vibrations, which cause this mucus. The ocean has a mucus film on top too, and your lungs do as well. And you get this rupture, and you get these aerosols that come out of people. And that was a big discovery during, actually it wasn't during, actually, I think I have it on the next slide. Yep. People wanted you to believe it, and I almost slipped and said it was. It was a discovery during, they kept saying, we're learning things as we go. But this was known back in 2009 that basically, that's cut off on there. So this is papers from Lydia Moroska's group. And basically it shows the size distribution of what comes out of humans. And it wasn't during SARS-2. We knew people produced aerosols, but it's been ignored, completely ignored by the medical community. And some people produce, billions of aerosols, and some people produce very few. It can change over the course of your infection. But yeah, so this is just, basically this was known. The question that existed was, do these aerosols contain viable virus, right? That was the question. That was actually someone who's here was involved in figuring it out and showing it, because they kept moving the bar of what was required. They kept saying, well, yeah, there's aerosols, but do they have virus in them? Is the virus infectious or not, right? They kept ramping up, moving the goalposts over and over. And, in fact, they said it doesn't count as airborne unless you can prove all that. Measles has never been sampled and proven to be infectious. And now SARS-2 has been, I don't know, we're probably up to, there must be 15 papers by now. Yeah, something like that. Yeah. Anyway, beautiful work, which we'll hear more about. All right. Okay. I try to talk to it. It doesn't help. It doesn't. Come on. Okay. So this is actually a figure from Lindsay Marr that she created, or actually her son created it. We had to pay him. I forget how much we paid him. Not very much. But basically, this is the current view of, what's happening. And, just basically said it over and over. Aerosols float like cigarette smoke. Droplets drop. And so that's where the 6-foot rule came from, was that if it's really mostly in the bigger droplets, which is what people expected before this pandemic, then if you were 6 feet away, they would just fall like cannonballs. Whereas you have to think of, if you're with an infectious person in a room and that person's smoking across the room, you might initially see them smoking, and then eventually you start to smell it. And if the room's poorly ventilated, then everyone can get infected. And that's what's happened. The interesting thing, or the hard thing, is that, some people would say, oh, it's not that infectious because I got exposed like 10 times and I've never caught it, right? But it turns out there's this little thing called the Pareto principle, which says that, it's for a lot of different processes, but in the case of this disease, 10% of the people led to 80% of the infections. So yeah, most of the time when someone's sick and near you, you don't get it. But if you are in a room with someone who's one of those super spreaders, everyone can get it. So anyway, so this is sort of the battle has been over this long range aerosol inhalation, which is the official definition of airborne, which they didn't like, but it's been shown now that that's pretty much can be the dominant pathway. Okay, this is an interesting story. How am I doing on time? How many? Okay, perfect, thank you. All right, so I gave a talk. I do grand rounds at hospitals now. This is crazy, right? They actually now, doctors didn't really like me at first, but they kind of grown to realize they have to put up with me. And so I just did grand rounds at our hospital on Friday, the pediatric hospital. And I said something on Twitter that I was going to be speaking there, and someone sent me this picture on the right, which is the messaging from my own university. I sent it to the chancellor too. And but this is the conundrum. This is the problem, and I hope we can talk about this at this workshop. Like, how do we get past this? You know, I've squawked. They know me. They run when they see me coming. But this is just unacceptable. It says hand washing is the #1 way to get rid of it, right? And so, basically, what are we doing after this much time? We know this is not true. This would be better messaging. And, this is actually a filtration that's now being put in more hospitals. The hard part about this particular virus, everybody wears the best N95s, and they, have filters, and they have the right rooms when they're with COVID positive patients. But if you sample in a hospital, most of the virus is found down the corridors, or in where the administration is, or in the elevator, or in the public restroom, or in the break rooms where no one wears masks, right? And so, basically, we need to change things and get people aware somehow, but I feel like they are aware and they're still not doing anything. So that's where I'm kind of stuck. There are fixes. This is one of my favorites. I've spent a lot of time putting Corsi Rosenthal boxes in schools. This little box costs $70 to make. So from a, sort of environmental justice perspective, everyone can have one. And it, you can make them cute little minions, and they outperform a HEPA by a lot. And so, basically a HEPA that could cost $800 is not sample. This can clean out a room of wildfire smoke, pollution, allergens, every respiratory virus in like 5 minutes. And so why does, we're trying to put these in all schools now. We have put them in one district and we're working on others, but it's completely cut back the spread. All right, I even took one to the White House. So, all right, the last little bit of time, I wanted to talk about air surveillance, because I know a lot of people talk about sewage surveillance. This is kind of sewage surveillance, but sewage surveillance in the air. And so, what can we learn about looking in the air and tracking disease? And so this is Jane Burns, one of my colleagues. I won't spend too much time because I'm running out of time. She's studied Kawasaki disease her whole life. It's the way that kids get #1 acquired form of heart disease. And this just shows Japan, San Diego, Hawaii, and wind direction, meteorological conditions. And when they line up, there will be Kawasaki disease cases across all of those different sites. Has to be the air. They're still trying to figure out what it is that's in the air. I call this a textbook example of air and disease. I don't know of a better one. So we've been looking in San Diego near a place where tons of massive amounts of raw sewage go into the ocean, called Imperial Beach. It is now a state of emergency. It has been, it's been there, this has been happening for like 40 years. The residents are rather angry. There's a broken infrastructure systems that basically on the Mexican side of the border that dumps into the US side. So it is not good. But basically we went out and sampled the air because they were only thinking about the water. And this is something I hope we can talk about more, is sort of this concept. Peter showed the slide of like human exposure has got like water going to a human, air, air going to a human, egg going to a human. But I expand that and think about water going to air to lots of humans, right? A lot more humans, sewage going to a lot more humans. We showed that if it goes into the ocean, gets trapped in the surf zone, it's like one giant toilet. It gets just massive bubbles, massive release of these pathogens into the air. We mimicked it with a pink dye that you can see down. If it escapes into the ocean, which it can do under certain conditions, you don't see it in the air, but if it doesn't, we see it. So we did the same thing for real pollution. We published a paper in March showing that up to 3/4 of the bacteria you breathe in the air in this region come from that river. So we've pinpointed exactly what it was through sequencing. You can see it comes out and it grows, and it's quite complex. Now we're doing a lot more with viruses. This year, and I'm sort of kicking up because I know I'm running out of time. This year was the year of all years. This year, as you know, we had a lot of rains, and more than I've ever seen in California. So when it rains, it's particularly bad, and those are images of what the ocean looks like. You can see 2023, the blue window is when we sampled. We did 42 days straight of sampling. So we captured the pathogens and the pollutants. We see methamphetamine, cocaine, all the different drugs as well in the air from this sewage. And, just cool, we saw phage, and they were intact. So that's a major indicator. That's like the best indicator. You ask about indicators. This is a great indicator of raw sewage. It took us a lot of work to be able to capture these things and for them to stay intact, but we finally can do it. And we're also, as I say, doing RNA analysis. All right, so the last thing I'll just say is these are the beaches. The colors refer to the bacterial levels. This was during Hurricane Hillary. There was sewage in the streets in Imperial Beach, California. And so, these beaches have been closed for 2 years. And so basically doctors in the area we've just started working with, they just looked at the number of gastrointestinal issues that people had during tropical storm. And you can see it's a huge increase. And the interesting thing is they asked them whether they'd gone to the beach or gone in the water, and most of them had not. So they were a little perplexed, like, well, how did they all get sick? It's in the air. That's my mantra these days. All right, so it's a problem everywhere. This is all over. There's New York, there's UK, San Francisco, especially after rains. We are dumping all of our raw and processed sewage into water and thinking it's gone, and it's not. It's coming back. And so I probably hope I didn't take up extra time. I was kicked in. I didn't. All right. I didn't have to talk that fast. Sorry about that. But yeah, so I'm going to stop there, and I think we're going to do questions now. Is that fine? Okay. Thank you very much.
Any questions here in the room?
I thank you so much. It's a very interesting talk and very thought-provoking. I'm wondering about wave action versus other mechanisms, right? So I'm from Louisiana. We have a lot of failing on-site sewer systems that are, enriching sewage in all these streams. And I'm curious about aerosolation in that context.
Yeah. So yeah, I focus a lot on waves. The other thing that will, lakes see this, lakes can even have waves. Rivers, you can see bubbles, right? When a river's, and when the water, when it gets, all of these systems, when you see it sort of dumping in, that's all, you can see it's falling a distance. That's all creating bubbles as well. So anywhere there are bubbles, there are aerosols. You would have to, and we were happy to help anybody who's interested in looking at those mechanisms. You would have to characterize those bubbles. That would help you understand sort of what's getting out, how much is getting out. But I guarantee you, if there's bubbles, it's something's getting out. And so we haven't looked at that. I don't know how different the bubble sizes would be, but it would be certainly something worth looking at, because yeah, as you say, like rivers, that's just another huge exposure pathway because they're going right through towns and stuff, right? So yeah, so I think that's a wide open question. I don't know anybody who has, I know people are doing it on lakes now, but I don't know anybody who's doing it in rivers yet.
I have a question about the trends that you show of the virus in air, but also infections, like those.
Was that the gastrointestinal?
The seasonal trends of the Japan disease.
Oh yeah. Sorry. Yeah. Okay,
so
go ahead.
Those are actual virus measured in air and also.
No, sorry, I went over that fast.
Okay,
thank you for asking me to slow down. Okay, so I'll explain it to you in normal speed. So sorry about that. No, so what those are the two things that were plotted were wind direction kind of, and number of Kawasaki disease cases. They don't know what it's due to. Okay. They have had, I don't know, 10 hypotheses of what they thought it was due to that it has been shot down. The hard thing on all of this airborne stuff, as I'm sure are aware, is there's a lag. So how do you build in the lag, right? That especially with, viruses, they can trigger something, you don't get the response
Yes.
till months or years sometimes, right after you've been exposed. So
Yeah.
that's, I think, what's getting them on Kawasaki disease. I will say that Kawasaki disease cases in San Diego went down when people were wearing masks.
Yeah.
They
And
went,
we observed that in Texas with RSV
yeah,
during 2020,
Yep.
during COVID, we
Yep.
saw those cases, and then
yeah.
Yeah.
I mean, it just doesn't, yeah, it just makes me crazy that we know it's such a fixable problem, right? And indoors, you know, it's insane. So the people in Imperial Beach are all putting air filters in their houses now, especially the ones that live on the beach. But I've kind of spoiled it for people thinking they're taking their wonderful fresh walk on Sunday mornings on the beach. They don't like me. They don't like my story.
We have a question from Esther
Oh, sure.
online. Please feel free to unmute.
Yeah, thank you so much. I'm a professor at Penn State, and my colleagues and I, first of all, thank you for the presentation. My colleagues and I have always had an interest in doing a comparison between like locations where you have low income housing with bad infrastructure in the US and similar places in the African context, for example. So as you're presenting, I was wondering, because many of the African places where we have projects, there's a high incidence of diarrheal diseases. How easy or how difficult would it be to do something like this that incorporates an African test bed?
It's a great question, and that's what I would love to do. My dream before I retire one day is to be able to do a global survey of, you know, what's out there. And, you know, now there's been some nice advances. I didn't talk about the sampling that we did. You know, there's been, one of the problems has been like, how do you help people sample inside their houses? You know, these pumps are noisy and whatever. Now there's huge high vol samplers that you need for bio in the air, or you don't see anything, that are really quiet, and you can just hand someone, and anyone can run them, and you can just, they're like little contained cartridges. And so there's ways you could essentially turn it into a citizen science project. That's what I want to do. And so I think it's very doable. We're doing this in Imperial Beach right now. We're giving it to people, giving them samplers to put in their houses. Yeah, it's totally doable. And you're absolutely right. I mean, the people that have been breathing the worst air, you know, their whole lives were the ones that got hit the hardest by COVID. And so we're trying to help them first, because there's so many things we could do. But yeah, it's a great idea. And if you want any, you know, if you want me to help you with samplers or information on that kind of side of things, you know, just reach out to me and I'm happy to share what we've done.
Thank you.
Yeah, thanks. I'm Jennifer Wiedis from the University of Utah. I have a quick question, and forgive my ignorance on the atmospheric chemistry side of this, but I live in a very dry area, and we have issues not with oceans, but with a lot of dust storms.
Yeah, absolutely.
So can you speak to what's known about pathogens associated with dust and transport from either local or international sources? Yeah,
and that's not a naive question. There's a lot more known about the dust than there is about sea spray. Sea spray was a bit of a surprise, honestly. But the dust, like, there's the best work that I know of, the most complete sort of record is Joe Prospero's work. If you look that up, he's looked at transport from Africa into the Caribbean and shown that things remain viable, bacteria remain viable. Nobody like comes over in the jet stream, right? The UV should kill it, but they hide, they nest inside of dust, and they use it to shield themselves. And so these microbes are smart. They've adapted. They've had a lot of time to adapt. And so anyway, so there's some really nice work by him and others. Now a lot of more people are looking at dust, but I don't think I think it'd be rare to find like naked dust that doesn't have something, you know, still attached to it, to be honest, especially fertile soils. And again, I know. And they showed, this is an interesting thing. They showed that when it comes from the most arid lands, like the harshest environments, those are the toughest ones. Those are the ones that somehow lived there. So flying through jet, you know, the jet stream is nothing, right? So it's kind of interesting. So the harsher the environment, they can actually survive there as well. Absolutely. As we get more and more, there's dust is everywhere. I studied, that's what I spent the first part of my career studying, was dust. There's a lot. It's everywhere all the time. Yeah.
Thanks for the talk. Really enjoyed it.
Thank you.
I'm curious, so with doing these grand rounds and talking with clinicians, how do you balance like inclusive communication? Because it's not just hand hygiene or filtration, but actually inclusive approach?
Yeah, it's a great question. I mean, I shamed them. I debated on whether I should show that slide taken in their hospital, but they took it well. You know, it's like I've tried the gentle approach that didn't work, so I took a bigger. You know, the hammers gradually got, and they're realistic. They push back. Actually, in that talk, one of the people said, well, you know, if you look back, rhinovirus, there was a really careful hand study done 50 years ago and showed that it came from not washing your hands. Okay, you know, rhinovirus, but that's also been shown to be in the air, right? It's just the ratio is different. So I will say, I was not the first to sort of say that it's like SARS-2 is 100% in the air. I think it's pretty close to that. But other viruses, they could be 50-50, or, you know what I mean? Like, we can't rule out surfaces for everything. But I will say that I do feel pretty strongly, and I don't know what others think, but I do feel strongly that I don't think you can get a pandemic from droplets, because you would not infect an entire, like, not everybody licks the same salad fork. Right. And so I feel pretty strongly that if you hear of major spread, and we're seeing this now for like dolphin packs and, wildlife. And when you see that, like, it's just a trigger to me that it has to be in the air, because we all share the same air.
Yeah, no, this is really great. Sam China from National Science Foundation. So my question has to do with surveillance, and what are the concentrations in the air? And obviously it's going to be different for outside and inside relative to what the current technology is for actually detecting what's there.
Yeah, it's low. It's really low, and in the air, and it's hard. And like RNA is even harder, because it just falls apart if you look at it, right? So we've worked really hard on trying to, you have to sample high volumes, but not do it in a harsh way. And we've spent years trying to do it well. I think the way that you'll hear maybe a little bit about their sampler, they actually coat it and then deposit it in a gel so it stays together. There's tricks you can play. The concentrations are incredibly low, but they're getting better. Like, we're just getting ready to start using cell sorters and being able to sort, and they can do sequencing on, in theory, one cell. So one virus or one bacterium. That's what we're moving towards. That's what we're pushing for. In air, we need that, and so the tools are getting there, but it's very, very low. We have to sample for a while usually, and that the longer you sample, the more chance you're going to destroy things, right? So it's a challenge. It's really hard. And so, but from a surveillance perspective, I didn't say this, but we're adding it to buildings. And the idea there is you can couple it with sewage. Sewage tells you there's somebody sick in that building. The air will tell you the weak point of the building. Like, is everybody getting sick in that break room, or is it the bathrooms or whatever, right? And we see, like in dorms, we see that it's mostly, we get hits almost every day in the elevators, because they like pull all the air from the whole building into them every time they move. And so, that's the nice thing about air, is you can start to pinpoint, where you could patch. It's not like you have to worry the whole building is bad, right? You can actually start to figure out the weak part of the system.
Oh, thank you for the presentation. I would like to ask you, what's your thought on the resuspension of these viruses in indoor environment? Like, we spend a lot of time in indoor environment, and somehow we do some resuspension activities, and there is not much talk about the resuspension of virus and the risk or the exposure from it. So what's your thought about it? Like,
in indoor environments, is that? Yeah. Yeah, I mean, they're going to be, viruses are usually, they're not alone, right? So they're attached to something. They could be in dust, as we were talking about. They will be resuspended. The question is whether, and I think this has kind of maybe helped us with SARS-2, is like whether they remain infectious and intact. They have to be intact, right, on a surface, and for how long. And luckily, I think for us, SARS-2 doesn't remain. It sort of likes the air better, but it doesn't really like surfaces. I think that's where we've gotten away with it. So the question is, yes, they will be resuspended. The question is, will they still be able to, do their attacks or not? And that, I don't think people know much yet, that I've seen anyway. I mean, we do it. It's really hard. We get stuck. The place we're stuck is answering the question of if it's infectious still or not. And because there's not that many BSL-3 labs where you have to go Ours is backlogged like crazy. So there needs to be NSF, there needs to be more, although NIH would probably be the one, but there needs to be more labs where you can do this because it's a limitation for us and everybody else right now. But what we do, like those images, that's the cheating way. If you can look at it and see that it's intact, then you can say it's more likely to be infection if you can grow it, but most things don't grow. But if you can grow it, that's another way. There's sort of ways you can check the infectious, but that's a hindrance right now.
I have a question about, so let's say we successfully in wastewater surveillance and air surveillance.
Yeah.
How do we really leverage it for mitigation at the building level, maybe at the community level?
Yeah, I think that's a great question. I think, you know, the first part is just sort of understanding, you know, it's just such a huge question, right? Like, where do you start? That's what I keep thinking. Like, where do you know? There's people have done stuff with like sick buildings, you know, but for this, you know, ideally, you'd like every building to be able to know that the air is clear, like, especially where people work, so you don't lose workers, right? There could be a pretty big financial incentive to doing this, right? But yeah, is in terms of like how do you get it implemented? I mean, there's different levels of implementation. Like I push these Corsi Rosenthal boxes; they're not the plan for the future. But what is starting to happen, like it's always been like chicken and the egg. Who drives it? So one of the things that's happening right now is different states are starting to enact indoor air standards, but then they're struggling with what metric to use, right? And so, because do you need, you know, we do this surveillance to see what's in the air, and I can tell you in daycare settings and in kindergarten, you see everything in the air, right? On every room you go into. And so, you know, and we know how to stop that. So why are we not doing that? And a lot of times they'll say, oh, it costs a lot of money or, you know, whatever. Like it's the information that this is the biggest frustration for me and everybody probably in this room is like, how do you can see I'm struggling with like, how do you go from just knowing what will work to fix it and fixing it? And we're stuck. And I think it's like climate change in a way. More people are interested in it. So you know, with climate, you know, I've always well funded, but with health, if I say the health word, the funding is like, it's comes out of this, you know, people are much more willing to listen to things about health and climate. Having said that, what's limiting climate and limiting this is misinformation and human choices, right? People are just choosing to move on without doing anything, right? Just pretend like it's gone and it'll go away. So I don't know. I mean, that to me, we were talking about who's in the room. Who do I think I want to sort of start talking to people like social scientists and people who think about choices for all this stuff. I think that's like, how did they get people to wear seatbelts automatically? You know, that kind of thing. We're at that point. I don't know if that didn't really answer your question, but yeah, okay.
That's really great. At NSF, we didn't try.
Yeah, exactly.
Bringing the social science.
Yeah.
Like there was a PIP for workshop was devoted to human PIP. I mean, they spend a lot of time discussing misinformation.
Yeah.
How do we nobody knows really
how it's been terrible.
Critical, I mean, just based on what you said. Right?
Yeah. So, yeah, it's been terrible. Yeah.
So anyway, there are many interesting ideas. You guys have it. Contact NSF.
Here's a question. One more. We have time for one more, right? Okay.
Oh, hopefully it's a good one then.
Yeah, hopefully.
I'm thinking kind of more broadly in how we can unite our exploration of disease transmission in aerosol much more into the arc of public health.
Mm-hmm.
Because initially this all goes back to miasma, where it was like bad air. But in reality, that during those time periods, the transmissions were primarily through bad food and bad water.
Right.
And so then
they think it was.
Well, for the cholera ones, those
yeah,
were kind of really
they turned off the handle, right? Yeah.
For it, and so then thinking about globally as our different risk perceptions, are we focused primarily in the United States based off of having a built infrastructure that allows us to concentrate on aerosol transmission? Where if we go to different contexts that don't have access to sanitation, where the El Tor pandemic is still going on for cholera and malaria is spreading in primarily tropical regions. How do we develop a comprehensive risk communication plan where it doesn't overweight one over the other?
It's a really good question. Yeah, I mean, I think, I don't know the answer, but it's a really, I mean, it's something we worth, I think, discussing. I'm curious what other people think. I mean, we are getting contacted. We're going to start working with India because they have wastewater exposed everywhere, right? And so they're on it. They came to me and said, have you thought about doing air surveillance? You know, they're already doing the wastewater, one group there through Tata. And so we're going to start helping them. So I'm trying, you know, I get pulled because I don't, you know, I know what's in my backyard, so I'm trying to fix that, right? But at the same time, that's nothing compared to other people in the world, right? And so I have a little bit of time to make a difference, help people, right? And who do you help first, right? Everybody needs help. And so that's where I go after the people. I feel very protected with the people that have been breathing the worst air the longest. You know, I almost feel guilty in some ways helping California because California is so far ahead and it's nothing. But I do feel good because I know the tools, and then I can take our tools that were successful and help other places. That's what I plan to do, and spend less and less time in my own backyard over time. But yeah, it's just so bad right now. And it's bad because information, I would argue, if they knew and they just, anybody I've helped put CR boxes in their house, like someone else in the house would have it, no one would, including our house. My son brought it home, locked him in his room, and threw a Corsi box outside his door and said, stay in there, and no one else got it. What a good mom I am. But yeah, I fed him too, kind of. I left food at the door. But anyway, no, but I think, you know, it's not like we don't understand. It's the same thing with climate. Like in some ways I feel like, do we need to do more? How many more studies do we need, right? We know what's there and we know how to get it out of the air. So, you know, we filter our water. I had that on Peter's slide. You know, we focus on, we wouldn't drink pathogen laden water, and we drink 2 liters of water a day, and we inhale 11,000 liters of air, and we have no problem inhaling pathogen laden air. It makes no sense.
Hi, thanks for the talk. I wanted to jump off of Mamadou's question about surveillance and making surveillance useful. And I'm in the waterborne pathogen world.
Okay.
And in that world, we have surveillance monitoring for a couple of reasons. And, you know, one reason is, you know, just health risk monitoring. And so we have indicator organisms.
It's an early indicator. Yep.
Well, not even the early, that's the second I was going to say.
Okay.
So even, you know, one thing is just like, is there a health risk from this water? We look at E. coli and total coliform.
Right.
Another thing is early indicators. And I imagine with air sampling, there's a similar thing, right?
I think so.
Early indication, but I'm wondering if there's any thought about that, just that health risk indicator, because it's so hard to look for viruses in air. Are there other things we could use as proxies just to say, is this room safe?
I agree. That's a great question. I didn't talk about it. I squeeze a lot in 30 minutes because that's my question. That's one of the things we do want to answer. And that's why we're using our little playground of Imperial Beach. It's not a playground for them, but we're actually trying to answer that question, right? They're using enterococcus counts, right? And as an indicator for the water when they release it. Even in the processed water, I would, you know, there's viruses, and you know what I mean? They're not looking for those things. And so that's the question is, what is a good indicator in the air? Is enterococcus, probably we see that in the air too. But we also see, I didn't talk about this, we're gonna have to talk about antibiotic resistant genes. We're seeing our number 2 most abundant bacterium is one that's known to do so this is also an antibiotic resistance issue. But what should you be, it'd be so nice if you knew what, like the equivalent indicator. I don't think it's probably enterococcus for the air, right? But what is it? And I don't know, you know, it's like, it's transforming your, I think now, like when I gave my rounds, one of the doctors just flooded me afterwards, by the way, and that's way different. And one of them was the head of IBD, you know, inflammatory bowel disease studies in children. She said they exploded in Imperial Beach this year, and they didn't know, these kids had never gone in the water, never gone to the beach, like, and so when she heard it was in the air, like, oh my God, they're breathing it in and ingesting, like that they have not even, most people haven't even thought of this pathway. So it's, again, what is the indicator? What is the health indicator? Like what could we, if we know what people are going to the hospital for, could we then, we have those air samples. So then can we go and figure it out? You know, it's just, there's so many things you could measure, right? But that's what it keeps me awake at night. So we have to figure that out. Yeah.
Okay, this has been a tremendous discussion, and I always hate to cut off a good discussion, but I'll do so with a promise. And that's, we're going to continue the conversation.
Absolutely.
So thank you so much.
Absolutely.
I just wanna
Thank you.
say Thank you again for this
Yeah. Yeah.
excellent presentation.